Greetings and welcome to the Journal Club. Today we are discussing a fascinating preprint that asks one important question for malaria drug development. Are our next generation malaria drugs already vulnerable? So this is quite an ambitious project where they're already looking at antimalarials, next generation antimalarials, and how they would behave potentially, and the resistance that could arise from their use. So we're going to explore how Plasmodium falciparum evolved an unexpected mechanism to survive exposure to one of the most promising next-generation peroxide antimalarials, OZ-439, also known as Artefenomel. We'll begin with why OZ-439 was developed in the first place, follow an extraordinary 476-day evolution experiment, then examine why conventional resistance assays failed to detect the phenotype, and finally discuss how metabolomic profiling uncovered an entirely different survival strategy that has important implications for resistance surveillance. So why develop next generation peroxide antimalarials? To set the stage, one of the greatest threats, as we all know, to malaria control is the continued spread of partial Artemisinin resistance. Much of this resistance is associated with mutations in the Kelch13 gene. First, it was identified in Southeast Asia and now emerging independently across multiple regions of Africa. To address this challenge, researchers developed a completely synthetic class of peroxide antimalarials known as ozonides. Like artemisinins, ozonides contain the essential peroxide bridge, and once this is activated by iron or heme inside the parasite, they generate reactive radicals that produce the widespread oxidative damage to the parasite. However, OZ-439, or this ozonide, was specifically engineered to overcome several limitations of conventional artemisinins. Its chemically stabilized trioxolane scaffold gives it a major pharmacokinetic advantage, whereas artemisinin derivatives have a half-life of roughly 1 hour. OZ-439 has a half-life of approximately 42 hours. This prolonged exposure raised an exciting possibility. Could a single dose provide sustained parasite killing while improving patient adherence? Because, if the patient has to take a drug multiple times, they may not remember. If they're starting to feel better, they may not take it. So if you can just give one drug that is longer acting than the hour or up to 6 hours that artemisinin is around for, then that helps. The authors wanted to have a look at whether parasites could eventually evolve resistance to this next generation peroxide. Instead of us just waiting and reacting, they've been proactive. So before we look into this new drug, Kelch13 is a protein we've all become very familiar with, because it's the molecular hub of artemisinin resistance. So to answer that question, we first need to understand the K13 protein. The Kelch13 is a substrate adapter within the parasite's ubiquitin proteasome system, and these ubiquitin proteasome systems are what cells use to degrade and recycle proteins. The main job here is to identify proteins that need to be removed or regulated. They tag it with a small protein called ubiquitin and then destroy them in a large protein complex called the proteasome. And this Kelch13 protein is a substrate adapter within the parasite's ubiquitin proteasome system. The validated Artemisinin resistance mutations cluster almost exclusively within the Kelch propeller domain. The well-known R539T mutation is one of those canonical resistance mutations. The mutation described in today's paper, A212T, is different. It lies outside the propeller domain within the coil region of the protein. Although no experimentally determined full-length structure of P. falciparum Kelch13 currently exists, Domain mapping clearly shows that A212 occupies a completely different structural region from the classical resistance mutations. This immediately suggested that the parasite may have evolved an alternative route to survival. To investigate this possibility, the researchers began with an artemisinin-resistant Cambodian parasite line carrying the K13 R539T mutation. They subjected these parasites to continuous OZ-439 selection for 476 days. Drug concentrations were gradually increased from 44 nanomolar to 1 micromolar, forcing the parasites either to adapt or die. Throughout the experiment, parasites' populations underwent whole genome sequencing. Candidate mutations were then recreated using CRISPR gene editing before detailed phenotypic characterization. If you're unfamiliar with the CRISPR system, it basically uses a guide RNA to direct a protein, usually Cas9, to a specific DNA sequence where the protein cuts the DNA. The cell's natural repair mechanisms try to repair the break, allowing scientists to disable a gene or correct a mutation or insert new DNA in this case. So after 500 days, nearly 500 days of continuous selection, a previously undescribed mutation emerged. By day 476, every sequence parasite clone carried K13A212T. Importantly, whole genome sequencing identified very few additional mutations, and A212 became fixed in every evolved clone. So this means that fixation, meaning that every single clone had this mutation. And so because every single clone had this mutation, it suggests that it confers a survival advantage under prolonged OZ-439 pressure. To determine whether A212T was truly responsible for the phenotype, the researchers reconstructed the mutation using CRISPR-Cas9. This generated isogenic parasite lines differing only at the K13 locus. And so when they talk about isogenic parasite lines, it means the parasite lines are identical except for one thing, which is this, one specific thing that they will be studying. And in this case, is the A212T. So the rationale is because the genetic background is identical, any subsequent phenotypic differences could be directly attributed to the introduced mutation. So phenotypic meaning what you actually observe. So a phenotypic trait is something like eye color, height, blood type, skin pigmentation. So once they saw this A212 mutation being consistently held onto, the edited parasites were then tested using the standard assays routinely employed to monitor antimalarial resistance. These included the ring stage survival assay and the conventional 72-hour IC50 assay. Given nearly 500 days of experimental evolution, one might expect these assays to reveal substantial resistance. But lo and behold, the assays failed to demonstrate this. The ring stage survival assay is commonly used in malaria research. It's used to measure how well very young ring stage Plasmodium falciparum parasites survive exposure to an antimalarial drug. We're talking parasites that are 0 to 3 hours old. And how they survive antimalarials such as Artemisinin or its derivatives. The ring stage of the parasites is used because it's been noticed that some tests fail to detect the type of reduced susceptibility that is there in some artemisinin-resistant parasites. So the ring stage assay focuses on the developmental stage where this resistance phenotype is most apparent. And so if you use parasites from other stages, you can mask the phenotype the assay is intended to detect. So if you use trophozoites or schizons, they may die, and then you think your drug is effective, when in reality, the ring stages are capable of resisting it. It turns out that artemisinin-resistant parasites often do not ignore the drug. They still get hit by the drug. But what happens is a fraction of early rings enter a temporary growth arrest state and later resume development once the drug is gone. And that's why it's really this stage that you want to assess the ability of the ring stages to revive once the drug passes through the body. So you're effectively looking at which parasites survive and recover rather than which parasites are immediately affected. So with this paper, they had an incredible shock because when they assessed the result, it turns out that the double mutant parasites showed no meaningful increase in OZ-439 survival during the ring stage survival assay. Meanwhile, they know they've driven resistance in this parasite by exposing it to the drug for 476 days. So likewise, there's another assay you can use. This is a 72-hour IC50 assay, and that looks at the inhibitory concentration 50. And this means once you put the drug, you want to know what concentration eliminates 50% of the parasites, essentially. And when they compared the IC50s at 72 hours, so that's like 3 days, post-treatment, they didn't find any difference between the resistant lines that they had created and the ones that should be susceptible. By every conventional metric, these parasites appeared fully susceptible. So this completely raises concerns about the assays we're using to test for susceptibility at this point. So sure enough, the researchers realized that perhaps they were simply observing the parasites for too short a period. So instead of ending experiments after 72 hours, they extended and followed up for 14 days. And this completely changed the story. What they found was that the double mutant parasites consistently recovered much earlier than the parental parasites after OZ-439 exposure. So what was happening? Rather than resisting the initial oxidative damage from the drug, they simply returned to growth sooner once drug concentrations declined. Importantly, the same pattern was observed with OZ-277, suggesting this was not unique to OZ-439, but may represent a broader vulnerability of peroxide antimalarials. So they didn't stop there. It's a phenomenal amount of work. They went on to examine parasite recovery in a humanized mouse model. Following a single oral dose of the OZ-439, the double mutant parasite recrudesced, that means recovered, several days earlier than comparison parasites. The in vivo findings mirrored the in vitro observations remarkably well. Together, these experiments demonstrated that the phenotype was biologically meaningful rather than an artifact of cell culture. So once they established the phenotype, the obvious question became, how are these parasites surviving? To answer this, the authors performed untargeted liquid chromatography mass spectrometry tandem MS, which is mass spectrometry, so you do it twice, metabolomic profiling. They compared ring stage and trophozoite stage parasites exposed to dihydroartemisinin, OZ-439, and untreated controls. This allowed them to identify global metabolic changes associated with the new mutation. The first observation was striking. The double mutant displayed widespread metabolic changes across multiple pathways. Rather than altering a single metabolite, the parasites appeared to rewire their overall metabolism. This suggested that survival was being achieved through coordinated adaptation rather than a simple biochemical change. Still, pathway enrichment analysis identified one pathway above all others. The glutathione metabolism showed the strongest enrichment in ring stage parasites. Levels of glutathione disulfide and related metabolites were significantly increased in the double mutant. This is consistent with enhanced antioxidant capacity and more effective buffering of peroxide-induced oxidative stress. But the story did not end with glutathione. Aspartate metabolism, pyrimidine metabolism, and nucleotide biosynthesis were also enriched. Together, these pathways provide reducing power, biosynthetic capacity, and the resources required for rapid recovery once drug concentrations fall. Rather than preventing oxidative damage, the parasite appears better equipped to survive it. So if we are to bring it all together, the data support a new mechanism of survival. So OZ-439 is still activated inside the parasite. Oxidative damage still occurs just as it would with artemisinin. However, enhanced glutathione metabolism, together with increased amino acid and nucleotide metabolism, allows the parasite to tolerate this damage. It enters temporary quiescence and recover much sooner after drug exposure. And this represents a fundamentally different survival strategy from classical artemisinin resistance. Importantly, or crucially, these findings challenge the way we currently monitor antimalarial resistance. Current surveillance focuses primarily on K13 propeller mutations and conventional phenotypic assays such as the ring stage assay and the IC50 measurements. Yet this study demonstrates that clinically important survival phenotypes may arise outside the propeller domain and remain completely invisible to standard assays. So future surveillance may therefore require broader sequencing of the entire K13 gene if it's not already being done. I'm not sure how those things are being tracked currently, together with longer-term assays capable of detecting delayed recrudescence. Thank you so much for checking out the video. I really appreciate you subscribing to our study group and giving the video a thumbs up. And I'm looking forward to catching you in the next video. God bless. Bye.